our love analysis technique. The study of phosphate solubilizing microorganisms, PSMs, as natural fertilizers in agriculture involves a range of analysis techniques to assess their potential effectiveness. These techniques encompass various aspects, from identifying and characterizing PSMs to evaluating their impact on plant growth and soil health.
Identifying and characterizing PSMs. The first step in utilizing PSMs is identifying and characterizing them. This involves isolation and screening. Researchers use various techniques to isolate PSMs from soil samples. This often involves culturing microorganisms on media containing insoluble phosphate sources. PSMs are identified based on their ability to form clear zones around their colonies, indicating phosphate solubilization. Molecular techniques. Modern techniques like 16S rRNA gene sequencing and metagenomics are employed to identify and characterize PSMs at the molecular level. This allows for a more comprehensive understanding of the microbial community involved in phosphate solubilization. Psychological and biochemical analysis. Researchers assess the psychological and biochemical characteristics of PSMs, including their growth rate, pH tolerance, and ability to produce organic acid and enzyme involved in phosphate solubilization.
Evaluating the effectiveness of PSMs. Once identified, PSMs are evaluated for their effectiveness as biofertilizers. In vitro studies, laboratory experiments are conducted to assess the ability of PSMs to stabilize phosphate in controlled environments. This involves measuring the release of soluble phosphate from insoluble sources in the presence of PSMs. Greenhouse experiments. Greenhouse trials are conducted to assess the impact of PSMs in plant growth in a controlled setting. This involves compassing the growth of plants inculcated with PSMs to control plants. Parameters like plant height, biomass, and phosphorus uptake are measured. Field trials. Field trials are conducted to evaluate the effectiveness of PSMs under real-world conditions. This involves comparing the yield and nutrient uptake of crops grown in fields inculcated with PSMs to control fields. These trials considers various factors like soil type, climate, and crop variety.
Analyzing the mechanism of phosphate solubilization. Understanding the mechanism by which PSM-soluble phosphate is crucial for optimizing their use as biofertilizers. This involves organic production. PSMs produce organic acids like gluconic acid, citric acid, and lactic acid that can dissolve insoluble phosphate compounds. The production and release of this acid are analyzed using various techniques like HPLC and spectrophotometry. Enzyme activity. PSM produce enzyme-like phosphates that hydrolyze organic phosphate compounds, releasing inorganic phosphate. Enzyme activity is measured using specific acidity. Microbial interaction. Researchers investigate the interaction between PSMs and other microorganisms in the soil. This includes understanding how PSMs compete with other microbes from nutrients and how they might influence the overall microbial community structure.
Evaluating the environmental impact of PSMs. Assessing the environmental impact of PSMs is essential for ensuring their sustainability. This includes the soil health assessment of researchers evaluate the impact of PSMs and soil properties like pH, organic matter content, and nutrients and ability. This assists whether PSMs improves soil fertility and reduce the need for chemical fertilizer. For water quality and analysis, researchers analyze the potential for PSMs to leach into water source and contribute to erosion, and they assess the impact of PSMs on water. And quality parameters like nutrients level and algal grow.
In our study have explored the mechanism of phosphorus solubilization processes in soil. Phosphate solubilizing bacteria (PSB) dissolve insoluble phosphate by secreting low molecular weight organic acid. In alkaline soil, phosphate precipitate to form calcium phosphate, which increase their solubility and decrease in soil pH. PSB increase phosphorus availability by secreting organic acid that cause a decrease in soil pH, leading to appearance of divalent and trivalent form of inorganic phosphorus. HPO42 and PO43 organic acid are produced in a periplasmic space by direct oxidation, and their release into an environment is accompanied by a decrease in pH.
Efficiency of solubilization depends on the strength and nature of the acids. Tri- and dicarboxylic acids are more efficient than monobasic and aromatic acids, and aliphatic acids are more efficient in phosphate solubilization. Gluconic acid and 2-ketogluconic acid are the most common solubilizing agents for mineral phosphate. Other mechanisms of mineral phosphate solubilization by microorganisms includes the production of inorganic acids and the secretion of chelating agents.
Future direction and application. The research of PSMs is ongoing with several areas requiring further investigation. Developing effective formulations. Researchers are working on developing effective formulation of PSMs that can be easily applied to crop and maintain their viability in the soil. Optimizing application techniques. Researchers are exploring optimal application techniques for PSMs considering factors like timing, dosage, and combination with other biofertilizers. Understanding the role of PSMs in sustainable agriculture. Researchers are investigating the potential of PSMs to contribute to sustainable agricultural practices by reducing reliance on chemical fertilizers, improving soil health, and enhancing crop yields.